Good morning, Como Park biology students. Mr. Adair here. It is January 11th, Monday morning. Hopefully, you guys all had a great weekend. All right, we're going to start this week off with learning a little bit more about genetics and chromosomes and alleles and genes. So, this presentation will be an ed puzzle where there will be some questions you need to answer as we go through the PowerPoint. All right, a couple things to think back on from last week. Number one, what are the two differences between somatic cells and gametes? You could pause the video here and answer the question. And actually, I'll probably add a question to this. Number two, what are homologous chromosomes? How are they different than sister chromatids? We'll talk more about that today. And where are genes found? So as you're thinking about these, I'm going to go through them as this is a warm-up just so you can check your knowledge. But somatic cells are body cells. In humans, 46 chromosomes. Gametes are sex cells, sperm and egg, 23 chromosomes, half as many. Homologous chromosomes are chromosomes that have similar genes, although slightly different. So for example, you will get 23 chromosomes from your father and another set from your mother, and each pair of the 23 will have a homologous chromosome set that will code for the same type of gene, for example, a gene for hair color or something like that. Sister chromatids are identical copies of the chromosome that will be made during gamete formation, and I will talk more about that today and tomorrow, actually. And then genes are sections of DNA. So they're like long sections of DNA, and they're found on your chromosomes. Okay? All right, real quick. Gregory Mendel, he's the father of genetics. He's the first person to come up with dominant and recessive. He was an Austrian monk. He failed out of teacher school. He bred pea plants in his garden, and he came up with some. He predicted the results of meiosis before we discovered chromosomes. He really wasn't recognized for his findings, but he's basically the one that figured out that how genes can skip generations and things like that. And we'll talk about that this week. Some of the vocabulary words you need to know for this unit will be, and this week, are genetics, the study of inheritance. Basically, where did you get the traits that you got? We've talked about this a little bit already. What is a trait? It is a distinguishing characteristic that you inherit, like eye color for a plant, leaf shape, in the animal kingdom, tail length. A gene determines your traits. It's found on the chromosomes. An allele is a different form of a gene. So you're going to get two genes from your parents, and they'll be similar but slightly different. So we call the different genes alleles. You could get a dominant allele or a recessive allele, and we'll talk about that today. So first things first, a dominant allele will mask the recessive allele. When I say mask, it means it kind of wins or overshadows or will be expressed. So if you get a dominant, let's say if you have brown hair color, for example, this is a simplistic, not real example. You received a dominant allele for that brown hair color. And we express these dominant alleles with the capital letter, in this example, capital letter B. Remember, you get two copies of the alleles, one from your father and one from your mother during conception. Recessive alleles need to be inherited from both parents to be expressed, and they're going to be written as lowercase letters. For example, little b, little b. All right, some more vocab words that will be very important as we go forward this week. Organisms that have two identical alleles for a particular trait are said to be homozygous. Homo means the same. So if you receive, or in this case of a pea plant, if you look at the picture to the right, and we're going to talk about Punnett squares, the picture to the right is called a Punnett square, and we'll learn about that more. But if you see the capital T and capital T are the same exact, they're both capital T's, so they're going to be homozygous. And because it's a capital letter, it'll be dominant. A lowercase t will also be homozygous because they're the same exact letters, lowercase t's in this example, but it's lowercase, so it will be considered recessive. However, if you get one capital letter, one dominant allele from a parent, and one recessive allele from a parent, then we're going to call that heterozygous. Hetero meaning different, one big, one small. And we're going to go through these Punnett squares in a little bit, but what this is showing us is that, actually we're going to get to that in a little bit, so I'm going to not talk about that right now. Genotype is the genetic makeup of the trait. What is in the genes? So we use these letters to represent the alleles, which are the genes. Really, there's not capital or lowercase letters inside of your body, inside of the cells. It will be a molecule that will either make proteins that express the trait or not, based on if it is dominant or recessive. So the letter, in this case capital T or lowercase t, is just the symbol that we use as students, scientists, and teachers to talk about these things. The genotype is the genetic makeup, and so we're going to write that as big T, big T, little t, or little t, little t. Now, these letters will be expressed and then show the phenotype. The phenotype is the physical trait, and in this example, either tall or short. This example, capital T represents a tall pea plant, and lowercase t would represent a short pea plant. So if you look to the picture to the right, we have a homozygous dominant pea plant that's tall, and we have a heterozygous pea plant that is actually also tall because the dominant trait will win. If we looked at both of these pea plants in our gardens, in real life, let's pretend, we would not know that one is big T, and the other one is big T, little t, unless we did like a genetic test. We would just know that they're both tall, so they must have at least one dominant allele. Okay? A capital letter represents the dominant allele for big T, tall. A lowercase letter represents the recessive allele for lowercase t, short. All right. We're gonna go past this a little bit. Well, Mendel basically said traits are inherited as individual units. Some traits are mask, others dominant versus recessive. Then the law of segregation, which says that organisms donate only one copy of each gene in their gametes, because during meiosis, those copies separate. And that's kind of what I want you to start thinking about. How does this relate back to meiosis and gametes? Organisms inherit two copies of each gene, one from each parent. All right, we're going to talk a lot about this slide in a little bit, but this is, if you look at the very first cell up here, it has 4 chromosomes total. 2 pairs, the longer blue and the longer red are going to be homologous chromosomes, and the shorter blue and shorter red are also going to be homologous chromosomes. So meiosis, each chromosome makes a sister chromatid, as we can see in the second one, and then it goes through meiosis 1 and then meiosis 2, and at the bottom we have our gametes that have, if you look, 2 chromosomes in each gamete, which would be half as many as the very original. If this was a human, we would have 46 in this top picture and then 23 in the gamete. That's kind of hard to draw on an image and prove the point, so this is a simpler example. And you can see the homologous chromosomes separated, and you see some blue and some red kind of combined. We're gonna, that's called crossing over, and we'll talk about that. It's another way we get genetic diversity in our gametes. So I will go through this image again on our whiteboard, our digital whiteboard, so you can see it more. So law of independent assortment, allele pairs separate independently of each other during meiosis, meaning they could go every time a gamete is made, it's random which chromosomes end up in that gamete. And this example only has 4 chromosomes. Imagine 46 all being randomly assorted. So every single sperm cell or egg cell is very unique to itself. And that's why, and we're going to talk more about that, why you are so unique genetically as well. And then different traits are inherited separately. All right, I'm gonna move on past that. Let's see, Punnett squares. All right, we're gonna learn a little bit about Punnett squares today. And I'm gonna draw pictures regarding this too to go over what a Punnett square is, and we'll go through a bunch of examples of this. A Punnett square, though, is that box that you've seen in this lesson, and it shows the possible gene combinations. It's like a way to predict if you know the genotype, which again is the letters like big T, little t. If you know the genotype of the parents, you can make a prediction of what the odds are that the offspring would get the type of genes in what combination. So, if you look at this picture, we have a tall pea plant on top and a tall pea plant on the left here. Big T, little t. I'm giving you the genotype. Big T, little t, big T, little t. When we make our Punnett squares, we're going to separate the big T on one side and the little t on the other. And I will draw this out for you later. In this case, separate the big T and then the little t. And then we combine them. So this is showing if this heterozygous tall pea plant mated with a heterozygous tall pea plant. Yes, plants sexually reproduce. There are male and female components. But anyways, that's besides the point. We can see the possible results of their offspring. So if this gamete joined, if the tall big T gamete joined this other big T gamete during sex, then the offspring would have a big T homozygous dominant. If the little t gamete joined the big T, we'd have big T, little t heterozygous. In this case, we'd have heterozygous. And then the last case scenario, if the little t gamete happened to join the little t gamete of the other plant, we'd have a short pea plant. So what we can say here is that 1 out of 4, or 25%, would be short. 3 out of 4 would be tall. 1 out of 4 would be homozygous dominant. 2 out of 4 would be heterozygous. Take some time to look at this, but if it's confusing, we're going to practice this a lot, so don't stress out. Probabilities predict averages. So the Punnett squares give us probabilities, and they predict the average outcome of a large number of events. Probability cannot predict the precise outcome of an individual event. So we're kind of making, what are the chances of this happening if we know the parents? In genetics, the larger the number of offspring, the closer the resulting numbers will get to be expected values. All right, so for now, I'm going to stop it there. I'm going to make a I'm going to make a ed puzzle to go through, so you will need to answer some questions based on our PowerPoint today. And then we're going to move on from there and actually do some practice Punnett squares, and I'm going to guide you through that process. So thanks for spending this time going through the PowerPoint today. Make sure you answer the questions, and they will automatically submit. And we will be in touch. Have a great day. Goodbye.